Bismillahirrahmanirrahim. Assalamu alaikum dear students. Today's lecture is about the medicinal chemistry of the analgesic. So in this lecture we will learn about some of the examples of the medicinal chemistry of the analgesic drugs. As you know that an analgesic is also called, the analgesic drugs are also called simply an analgesic or pain reliever or pain killer. So these are the classification of the analgesics. So this we will cover some of the examples of the medicinal chemistry of the NSAID. And then after this we will see some of the examples of the opioid, which is also called the strong analgesics, and then we will see the some of the examples of the some of the local anesthetics. So first we will go for these non-steroidal anti-inflammatory drugs, and then we will cover these opioid analgesic and then the local anesthetics. So for what is important in case of the non-steroidal anti-inflammatory drugs are the inhibition of the cyclooxygenase. So that what is the primary function of the non-steroidal anti-inflammatory drug is to inhibit the cyclooxygenase or the prostaglandin synthesis. So this is you can see these are the structures of the prostaglandins, right, which is composed of the 20 carbons, and which include the cyclic ring structure. So what generally these non-steroidal inflammatory drugs did is it inhibits the cyclooxygenase, and there is no further synthesis of the prostaglandin. So so the prostaglandin is actually the extremely powerful chemical which is which perform the job at very low concentration. So it is the prostaglandin is mediated some uncomfortable and unpleasant body processes, for example the pain and inflammation. So there are some of the descriptive or romantic sounds of the prostaglandins. So some of the prostaglandins, which is PGD2 and PGF, and then PGE, and these are some of the prostaglandin. So what generally the function of the NSAID is it inhibits the cyclooxygenase. So there is no further conversion of this arachidonic acid into the prostaglandin. So that's how at this stage the NSAIDs are bind to the cyclooxygenase to avoid the binding of arachidonic acid, which convert this the arachidonic acid into the prostaglandin. So these are some of the examples of the NSAIDs, the aspirin, ibuprofen, and so what these drugs are actually classified as the arachidonic acid pathway inhibitor, right? So it inhibits the synthesis of the prostaglandin via the conversion of the arachidonic acid into the prostaglandin, right? So this is somehow the synthesis of the aspirin, which have been performed in 1897. So this is the acetylated form of the salicylic acid. So this is salicylic acid. This is anhydride acetic acid, which in acidic condition convert into the acetyl salicylic acid, which is also called the aspirin. So actually what is the mechanism of the NSAIDs? Actually they act as a competitive inhibitor. So what they generally do is they inhibit at this stage, right? So they compete with the arachidonic acid for the cyclooxygenase binding, so that the arachidonic acid cannot bind to the cyclooxygenase and cannot convert this arachidonate into the prostaglandin. That's how the NSAID performs its function. So there are some of the isoform of the COX. For example, the COX-1, which is the non-selective, COX-2 is the selective, and these COX-3. So these are the some of the isoform of the cyclooxygenase. So COX-1 and COX-2 are these selectively inhibited by these anti-inflammatory drug, while the COX-3 is actually inhibit the analgesic or antipyretic drugs. So these are some of the functions how and where the COX-1 express. So COX-1 is expressed in the most cells, which it also described as a housekeeping enzyme, which are also regulating the normal cellular function. For example, the gastric cytoprotection and vascular homeostasis. So the COX-2 is actually these expressed in the brain, in kidney, and in the bones. So what is the difference between the COX-1 and COX-2? Actually, COX-1 has the isoleucine at the position of 434 and 532. So you can see this isoleucine in the COX-1, and in COX-2 there is a vanillin. So vanillin. So this is only the difference between the isoleucine and this is the difference between the COX-1 and COX-2. So you can see this COX-1, which acts as a non-selective COX inhibitor. So this how the compound inhibits. So you can see there is a difference between the COX-1 and the COX-2. In case of the COX-2, there is a vanillin, and there is a COX-1, there is isoleucine, and this is how the compound binds with the COX-1 and inhibit the function of the COX-1, and the same way it binds to the COX-2 and inhibit the function of the COX-2. So now we come to the classification of the analgesics. So these are some of the classification. For example, this salicylic acid derivative, para-amino phenol derivative, which is acetaminophen, and this indometacin from the indole and indene acetic acid. So these we will see some of the examples of these classified analgesics. So first we will come to the salicylate, and then we will see some of the example of the aryl-alkylonic acid, and then these aryl-and thenolic acid, and then come to the selective COX-2 inhibitor, and then some of the example of the COX-3 inhibitor. So this is the salicylate. You can see this is the salicylic acid, and these are the derivative of the salicylic acid. The acetyl salicylic acid, which is also called the aspirin, and then these salicylamide. So and then salicylate. So you can see these all the compound which is marketed the derivative of the salicylic acid, which acts as the NSAIDs. So what is common in case of the salicylate is they can reversely bind to the cyclooxygenase COX-1 and COX-2, right? And then salicylate also inhibit the platelet formation of the lower cardiac mortality. So structure activity relationship is in case of you can see in all the compounds the most important thing is they all have the carboxylate anion. So you can see the carboxylate anion. So all compounds contain the carboxylate anion. And then except this the salicylate amide. Salicylate amide has no carboxylic acid, no carboxylate anion. That's why it has analgesic action but lack of inflammatory action. So there the substitution on the carboxyl group or hydroxyl group change the potency or the toxicity of the salicylic compounds. For example, you can see in case of these compound where is the substitution at the hydroxyl position, for example the acetyl group, for example this salicylate group, right? So it can also differ the potency or toxicity of the salicylate compounds. So the most important thing is the presence of the carboxylate functionality in order to act this salicylate as a COX inhibitor. So the structure activity relationship in case of the salicylate. So you can see the the active moiety appear to be as the salicylate anion, which is this one, salicylate anion. And then the side effect of the aspirin, for example, this GI effect appear to be associated with the carboxylic acid functionality. So if, let's say, for example, reducing the acidity, if we convert this compound into the amide, so for example, in case of the salicylate amine, it maintains the analgesic action of the salicylic acid derivative but eliminates the anti-inflammatory property, as you can see in case of the salicylate amine as compared to the salicylate. So if, let's say, the substitution on either carboxylic acid, for example, this one is the carboxylic acid, or the this phenoxyhydroxyl, which this is phenoxyl hydroxyl, right, may affect the potency or affect the potency and the toxicity. So the most important functional group is the carboxylic functional group as well as the this hydroxyl group. So benzoic acid itself has a weak activity. So placing phenoxylic hydroxyl group at meta position or para position to the hydroxyl can abolish the activity, right? So that's so that's why this phenolic group or the benzoic acid group has the weak activity. So if the substitution of any halogen compound, for example, chlorine, bromine, iodine, right, also on the aromatic compound which is substituted at the para position of the hydroxyl group, right, it can enhance the potency and the toxicity. So, and the substitution of the aromatic compound at 5 position, which is in case of the diphenyl, right, increase the anti-inflammatory activity. In case of the aspirin, it is acetyl derivative of the acetyl derivative of the salicylic acid. So it usually it has a unique amide, unique acid. So it covalently bind with the isoform of the COX and thus inactivate them irreversibly. Right. So you can see this aspirin, and this is the prostaglandin. So that's how you can see this one aspirin is bind with the this prostaglandin, and it can acetylate the prostaglandin, right? That's how it acts as act in a unique way as a COX inhibitor. So these are some of the metabolic fate of the salicylic acid. You can see gluconide derivative. In case of this gluconide derivative, the gluconization at this hydroxyl group, or the aspirin convert into a salicylic acid, and then further it can be gluconidation or this glycine and silyl transferase. So these are some of the salicylate derivatives which convert into the salicylic acid, and which further be convert or gluconidation, or the further fate is the glycine. So these are some of the metabolic fate of the acetyl salicylic acid derivative. And then the second example is the this aryl alkylonic acid. So this is aryl group, right, and this is the alkylonic acid group. So they act as a non-selective inhibitor. So the most example is aryl or heteroaryl acetic acid, in which these drugs come into this function. This group is the indometacin and diclofenac sodium. So which have the aryl group, or the heteroaryl group, right? And in case of the aryl and heteroarylpropionic acid, the examples are ibuprofen or naproxen. So it has aryl group, right, and the heteroarylpropionic acid. So this is propionic acid 1, 2, and 3. So these all comes under this aryl or heteroarylpropionic acid. So these are some of the structures of aryl or heteroaryl acid derivatives. This indometacin, diclofenac, and Sulindac and E-Sulindac, and these are all the examples of the heteroaryl acetic acid derivative. So this the first example is the indometacin. So you can see the indometacin, it contains this heteroaryl acetic acid group, right? So the preferred conformation in case of the indometacin is the generally the cis conformation. Cis conformation is the most stable one because it it has a less steric hindrance, right, due to the 2-methyl group, which is the 2-methyl group, right. Here is a more steric hindrance in case of the trans conformation of the indometacin. So kindly remember the cis conformation is more stable as compared to the trans due to the 2-methyl group at the indole group. So the selectivity relationship in case of the indometacin is right, the replacement of carboxylic acid, or if let's say we can replace the carboxylic acid with some other acidic functionality, it can decrease the activity. Means the carboxylic acid is the most essential group in case of the activity of the indometacin, right. So and then the amide analog are inactive, right. So if let's say convert the hydroxyl into amide, so it will be inactive, right. If let's say the N-benzoyl derivative, so N-benzoyl derivative, this is N-benzoyl derivative. If let's at the para position, if there is a para and halogen group at the para position, for example chloro, bromo, or trifluoromethyl, or the thiomethyl group, so these compound will be most active compound. So and at the fifth position of the indole functional group is most flexible, right. So this is the fifth position of the fifth position of indole group is the most active position, right. So 1 2 you can see 1 2 3 4 5 position which contain the these methoxy group, which is responsible to enhance the activity. So the most important functional group is the carboxylic acid, and then this one N-benzoyl derivative, and the fifth position of the at the fifth position of the indole, which is the most flexible group part, and which is most responsible for enhancing the activity. Now these are the metabolism of the indometacin. You can see that this indometacin convert into the glucuronide derivative, or some of the derivative which have the conversion of this methoxy group into hydroxyl group, right, and further the cleavage of the group, and then it convert into the glucuronide derivative. So structural activity relationship of the second drug, which a second drug which is the Sulindac, right, it convert it it is also the COX-1 inhibitor, right. So if let's say the substitution of the methoxy group or fluoro or dimethylamino or methoxy group, for example in case of the case of this there is a fluoro group, right. If let's say dimethylamino group or methyl group, it can be more active than unsubstituted mean. So meaning that the substitution is very important in case of the Sulindac. The presence of indole group, indole ring, is not essential for activity because the corresponding 1-benzyl diene analog, which is in case of the Sulindac, are more the active. So alkyl groups are especially, for example, the methyl group at the alpha position of the alpha position. So it is more active than the aryl position. So meaning that alkyl group is responsible for the activity. And the substitution of the methyl group at alpha position of the acetic acid, right, give the corresponding propanoic acid and lead to the equivalent analog. So this if let's say the substitution of this alkyl group at alpha position of this acetic acid to give the propanoic acid, which is also have the equimolar equiactive analogs. So this is the fate of the Sulindac, which is can see this converted to the Sulindac into the sulfone derivative, right. and then further the fate of this sulfone into the gluconide derivative. So these are some of the active metabolites of the Sulindac, which is resulted as a fate of this Sulindac metabolism. So in case of the difluronic sodium, you can see this diclofenac sodium can possess the three possible mechanisms. It can inhibit the arachidonic acid cyclooxygenase system, and it inhibits the lipoxygenase pathway and inhibit the arachidonic acid release. So the structure activity relationship in this compound is diclofenac is the presence of this chloro group, 2 chloro group at this ortho chloro group, which force this alanine-phenyl ring. So this is alanine-phenyl ring, right, out of the plane of the acetic acid, out of the plane of this acetic acid, and this twist effect being important in the binding of this compound of the NSAID to the active site of the COX. So meaning that this ortho position chloro group is most important one, which is responsible for binding this compound with the active site of the COX. So this is some of the this is the metabolism of the diclofenac. So you can see the fate of this diclofenac into the 4-hydroxy major metabolite of 5-hydroxy. You can see the hydroxyl group, and then the hydroxylation at 3' hydroxyl group, and then further metabolism of this 5-hydroxy group into the 4,5 dihydroxy. Another example of the analgesic drug is Nabumetone, which is the new class of the non-acidic prodrugs. It is the classic example of the prodrug approach in the drug discovery. This compound or this drug is 13 times more potent than the aspirin and 1/3 more as active as Indometacin and half as the active as the diclofenac. So this is the fate of the new Nabumetone. So you can see these convert into active metabolite, which is the carboxylic derivative of carboxylic derivative. This one you can see this acid, this ketone, this carboxylic acid, and another metabolite is the hydroxyl, which the methoxy convert into the hydroxyl, and then further the conversion of this hydroxyl into the reduction of the carbonyl group into hydroxyl, and then the glucuronide. So these are some of the fate of the Nabumetone. So the structure activity relationship in case of the Nabumetone is the introduction of the if there is introduction of the methyl or ethyl group on the side chain of the butanone, so it will reduce the greatly reduce the anti-inflammatory activity, right? And then second, if let's say conversion of this ketone functionality into the diazolone. Actually there is a oxygen group, 2 oxygen group in the ring, right? So if let's say the conversion of this ketone into diazolone will retain the activity, while if let's say the conversion of ketone into oxime is actually the carbon with the double bond N group and OH, right? So this is the oxime, which reduces the activity of the molecule. If removal of these this methoxy group, which is at 6th position, which will reduce the activity of the compound. So if there is a replacement of the methoxy group with methyl or chloro group, will produce the active compound. If replacing this methoxy with any hydroxy group or acidoxy group or N-methyl carbamyl group, or the position isomer of the methoxy group at 2 or 4 position greatly reduce the activity. So meaning that this subgroup, this part of the molecule is very important, and at this part the methoxy group play a very important role. So these are some of the examples of aryl and heteroarylpropionic acid. So you can see this this aryl group, and this is the propionic acid 1, 2 and 3 carbon. That's why it is come in under this heteroaryl propionic acid. So these are some of the examples. We can see the medicinal chemistry of the ibuprofen, naproxen, right? So for the ibuprofen, it is the first NSAID introduced since the Indometacin, and the first aryl propionic derivative to be marketed in US. So it is marketed in this as a racemic mixture, although the biological activity resides exclusively with this S isomer of the ibuprofen. So stereochemistry associated with the chiral carbon. This is the chiral carbon of this aryl propionic acid, right? But the linking in the acetic acid derivative play a very important role in both in vivo and vitro activity of this compound. So this chiral carbon play a very important role in the activity of the molecule. So this is this is the fate of this ibuprofen. Convert ibuprofen into active metabolite, right? So you can see it convert this part of the molecule into the hydroxylated tertiary butyl hydroxylated group, right? So these are some of the other fate of the ibuprofen. So the medicinal chemistry of or a structure activity relationship of the naproxen. This is the structure of the naproxen, which is consists of the 2 naphthyl acetic acid. So it is substituted in 6th position, lead to the maximum anti-inflammatory position because of this methoxy group. So if let's say, for example, the small lipophilic group such as the chlorine and this methyl sulfide or this CHF2O were active analog with the CH3O methyl being the most potent one. So if let's say these compounds substituted here, which also lead to the formation of the active metabolites. So large group found to be less active at this position. So derivative of the 2-naphthyl propionic acid more active, corresponding to the acetic acid, means the propionic acid play a very important role in this series of the compound. Replacing the carbonyl group with other functionality, for example, this ester group and then aldehyde group or alcoholic group will lead to the retention of the activity. Naproxen is only the alkyl glucuronic acid NSAID which marketed as a optically active isomer. These are some of the examples of N-aryl-alphanonic acid. So for example, so this is the example of this mefenamic acid is from the N-aryl-alphanonic acid. So this is the alphanonic acid, right? So these some of the specific drugs are mefenamic acid or the meclofenamate sodium. So the structure activity relationship of these compounds or this class of the drug is a substitution on the anthranilic acid ring generally reduces the activity. So meaning that if further substitution on anthranilic acid, this one, right, will reduces the activity, where substitution of the N-aryl ring can lead to the conflicting the result. So if the substitution here can lead to the more active metabolites. 2' chloro group, for example, in case of the meclofenic acid, appears to be essential for the activity, because the replacement of the NH functionality, for example this one, with oxygen, CH2 or sulfur dioxide or these NCH3, right, will reduces the activity, meaning this in case of this compound, this NH is play a very important role. So anthranilic acid derivative are active, whereas these meta and para aminobenzoic acid analogs are not. So meaning that the anthranilic acid, if let's say if ortho derivative is more active as compared to the meta and para aminobenzoic acid. Replacement of the carboxylic acid functionality with the tetrazole moiety has little effect on the activity. So if let's say this carboxylic acid replaced with tetrazole will little affect the activity of the compound. So now these are some of the examples of the COX-2 inhibitors. Salicylate, Refigozib. So these are some of the examples of the selective COX-2 inhibitor. So this is the metabolism of the salicylate, which is converted into this the hydroxylation at this methyl position, which convert this hydroxylated derivative, and further the hydroxyl will convert to carboxylic acid and then to gluconoid derivative. So this metabolism occurs in the liver by the cyclooxygen of this cytochrome CYP to C9 and involve the hydroxylation of the 4-methyl group. So this is the binding models of the between the COX-1 and COX-2. So you can see this is the example. This is the example of this Indometacin, right? And this is the example of the Selective, right? So these are the functional group which bind with the amino acid of the group of the COX-1, and here so these are important functional group. For example, double bond binding region, this one, and then this aromatic bind with the tyrosine and leucine. So these are the different functional group which bind with the amino acid of the COX-1, and the same in case of the COX-2 binding model of the Salicylate. So there are some selective examples of the COX-3 inhibitor, which is the Salicylate and phenacetin and this acetaminophen, right? So this is, you can see the in case of the acetaminophen, the inhibition of the or affinity toward the COX-3 is very high as compared to, you can see in case of the Indometacin, the Indometacin affinity is toward COX-2 is more as compared to the COX-1, and very less affinity toward this COX-3. So these are the comparison of the binding constant or IC50 value in millimolar of the selective acid for the COX enzyme. So metabolism in case of the acetaminophen, you can see this the acetaminophen, right? So phenacetin was the conversion of the phenacetin into the acetaminophen and then the gluconoid or sulfate derivative, right? Or the acetylite, which is converted into the acetaminophen, or the manner product is the aniline group, right? So this is and then the phenacetin, right, it is converted into the hepatotoxic compound, right? And then after this conversion of this oxidation at the nitrogen site, conversion of this into hydroxylated derivative, right? So this is somehow the gluconoid or derivative or gluconoid derivative or sulfur derivative of acetaminophen. So O-gluconoid being the primary metabolite in ethyl, while O or this O-sulfate conjugated being the primary metabolite in the children.